everyone!
So now, we're gonna discuss about the 7 prepared specimen on Microscopy 1.
So now, let's proceed with the introduction. Microscopy helps us observe the small structure of plants and algae. In this activity, prepared slides such as frame leaf, visual root leaf, exoral leaf, monocot and dicot samples, pinus cone, and fucus were examined to identify tissues and compare their structure. This helped us understand plant organization, reproduction, and diversity.
So for the objectives, compare structural differences between major plant groups, pteridophytes, gymnosperms, monocots and dicots, and algae, and relate those differences to function. Produce accurate microscope drawings, photomicrographs, and write concise captions noting magnification and identified structures. Record observations systematically, sketches, measurements if possible, and notes, and draw conclusions about form-function relationships and evolutionary adaptation.
In methodology, we began by wearing proper safety equipment such as lab gown, hairnet, face mask, and gloves. Each specimen was observed based on its color, shape, and other visible features. After viewing, our group discussed the results and assigned specimens to each member for detailed descriptions to improve the accuracy. Some members also gathered additional information from reliable online sources, and all of the findings were then compiled into our PowerPoint presentation.
Pteridophytes fern leaf XS with sporangia. At scanner view, the image captures the basic outline of the fern leaf and the sorus. For the LPO, or the inside of the sporangia, numerous spores can be observed, representing the reproductive cells of ferns. For the HPO, or high-power objectives, the internal cells appear more enlarged, showing rounded parenchyma and vascular tissues.
Cropped visual root chip. Under scanner, we can view the entire cone-shaped chip, including its protective root cap. Under low power, individual cells starts to appear as tiny dots arranged in rows, especially near the root cap, which indicates the zone of. And lastly, under high power, the cells look rectangular and compact. Some are undergoing mitosis, which is why the root tip is called meristematic region where continuous growth happens.
Pteridophytes fern rachis cross-section scanner. The fern rachis will appear very small and less detailed. You'll be able to see the overall structure, but finer details will be hard to make out. Power will appear larger and more recognizable. You observe the arrangement of cells and tissues, such as vascular bundles and ground tissue. Power, even larger, providing the most detailed. You can see cellular structures like xylem, flow cells, and epidermal cells with possible
Exoral leaf CS. Under scanner power objective, the whole exoral leaf cross-section is visible with its lamina and midrib. Showing only the general outline and symmetry, it looks like a spider abdomen. Under low power objective, the midrib and tissues are clearer. The epidermis, mesophyll, palisade, and spongy, and the vascular bundle, xylem and phloem, can be distinguished. Under high power objective, individual cells are detailed. We can tell they are parenchyma, mesophyll, but not confidently identify it. B311 monocot and dicot stem CS. Under scanner displays the entire cross-section of the stem. You can clearly see the epidermis, the outer layer, the cortex, and the vascular bundles scattered across the ground tissue. Under low power, shows a clearer view of the vascular bundles scattered throughout the stem section. These scattered vascular bundles are a typical feature of monocot stems. Monocot stems usually have vascular bundles arranged in a ring. Under high power, zoom in on the internal details of the vascular bundles at this magnification. You can observe xylem and phloem tissues more distinctly, although the clarity is slightly reduced in this capture.
Phaeophyta fuchus juchus male cross-section. Scanner. The outer region shows a distinct epidermal layer. The LPO, the arrangement of tissues, epidermis, cortex, and medulla, and conceptacles is clearer than under the scanner view. HPO saw some slight greenish tinge, which could be related to the presence of chlorophyll or other pigments in the algae cells. Phaeophyta fuchus juchus male cross-section. Scanner. The outer region shows a distinct epidermal layer. The LPO, the arrangement of tissues, epidermis, cortex, and medulla, and conceptacles is clearer than under the scanner view. HPO saw some slight greenish tinge, which could be related to the presence of chlorophyll or other pigments in the algae cells.